So now in this next video, we're going to jump right into a real-life DNA technology technique known as DNA sequencing. Now we're going to entitle this next flowchart DNA sequencing and look at a specific type of DNA sequencing that's used to give us some very valuable information about a DNA in question. Specifically, DNA sequencing is going to be utilizing a principle that we've already established. When you DNA sequence, when you try to figure out the sequence of DNA, what you're doing as a scientist is you're actually using the principle. So DNA sequencing itself uses the principle of something we've already established, something we already know, and that principle is complementary base pairing. It uses the principle of complementary, so we'll say COMP BP-ing. We know that A binds with T and vice versa. We know that G binds with C and vice versa. This is incredible and incredibly powerful knowledge that can help us do the following. This is the end-all be-all goal of DNA sequencing, which would be use this principle to extrapolate and sort of use that information to determine an actual gene's sequence. So we use this principle of base pairing to determine a real-life gene sequence, so a gene's sequence. And this is going to be very, very important in technology today. Now, before, this actually used to be a very laborious process. Now it's most of the time automated by sequencing machines that are devoted to doing this following principle and determination. So we'll say it's automated by sequencing machines. But what we're going to be looking at right now is a technique that still is done in many labs across the world that is a genius way to determine a gene sequence utilizing the principles of complementary base pairing. That technique is going to be known as the following. It's a long name, but we're going to shorten it, obviously. So it's called dideoxy. So this is a little bit different than what we've seen before. Ribonucleotide. So ribonucleotide chain, let me finish, nucleotide. So dideoxy, ribonucleotide, chain termination sequencing. Very, very long name for a very complex process. So, from this point forward, I'm not going to be saying dideoxyribonucleotide over and over again. I'm actually just going to say dideoxy, and that's usually the convention that most scientists use. Whenever they say dideoxy, they mean dideoxyribonucleotides. Just like when you say DNA, you mean deoxynucleic acid, and RNA, you mean ribonucleic acid. There's a shorthand for dideoxyribonucleotides, and it's just dideoxy. This is a technique that's going to utilize something known as chain termination. We're going to get to that, but first we're going to sort of lay out what this is going to provide us. Dideoxy chain termination sequencing is going to allow us to figure out about 1,000 base pairs, meaning that it's going to allow us to gain the knowledge of about 1,000 base pairs to be sequenced. We're going to be able to determine a gene sequence up to about 1,000 base pairs. So that's a very powerful tool. Now, the name itself, dideoxyribonucleotide chain termination sequencing, the specific part of the name that is sort of important to understand is the dideoxy part. This is based off of, just like DNA and RNA, the name of DNA and RNA and dideoxyribonucleotide is based off of sugar. So we'll say name based on the sugar that's present within those respective molecules. In RNA, the sugar is what? R stands for ribose, right? So ribonucleic acid is RNA. Ribose is the sugar. In DNA, we actually have two options. You know one of them because we've talked about it. One of them is a deoxyribose, right? So it's a deoxyribose. And why is it deoxy? That is because it's missing something. It's a deoxy, so it's missing oxy at the 2' position. So we'll say it's missing specifically what is known as a 2' OH. That's why we call it deoxyribonucleic acid. That's not new information to you. But what's new to you is the idea of a dideoxyribose sugar. This would be a sugar that is deoxy at 2 positions, meaning that it's actually missing an OH group at both the 2' OH and the 3' OH positions. So at the 3' it's missing an OH, and at the 2'. This 3' missing OH is going to be critical in this termination sequencing, okay? Just from this point forward, whenever you think of termination, associate it with the idea of a missing 3' OH. That'll make a little bit more sense now that we actually establish and sort of get to talking about the real process behind this complex sequencing technique. So let's look at the actual process that scientists undergo. First step is to do the following. You have a double-stranded DNA and you want to make it single-stranded. The term used here would be to denature DNA. Now I'll explain why in just a second. We're going to denature DNA, and the reason why is because we want to obtain out of this denaturation process a single strand. Okay, we just want a single strand for right now and serve as our template. What we're trying to basically figure out is the base pairs that this single strand has on it. Now, in order to figure that out, it makes sense for us to separate the double-stranded DNA molecule because we want to work with just one strand for simplicity's sake. It's going to be very difficult to figure out the sequence of a double helix structure. Let's just separate them utilizing this denaturation. So we're going to separate two strands and just get one single strand that will serve as a supposed template of sorts. We're going to take this single strand now, and as a scientist, we're going to literally put it in a, let's say, a tube of sorts with. So you're basically going to combine single-stranded DNA that's going to serve as a template and mix it up with a couple of different factors. You're going to mix it up with a bit of primer. You're going to mix it up with a very important enzyme that you know very much about, DNA polymerase. You're also going to mix it up with 4-deoxy. So 4 meaning that all 4 of the deoxynucleotides, okay? And you're also going to mix it up with, this one is a bit more critical, 4-dideoxynucleotides. So let's go over each of these. A primer is there simply because DNA polymerase needs some help. Remember, we're trying to determine a gene sequence. We know that there's a single strand. We basically want to use complementary base pairing to figure out what is going to be added onto the single strand if you mix it with DNA polymerase. What will DNA polymerase add on to the single strand? If you can figure that out, you've thus figured out the gene sequence because you know about complementary base pairing. You know that's just the opposite. So, DNA polymerase needs a primer. Specifically, this primer will just be, just like we've established before, a short stretch, so we'll say a short stretch of nucleotides of base pairs that are actually known to the scientists. These base pairs are actually already known to the scientists based off of the 3' end of our template. Known 3' end of our template. So this template, this single strand, scientists have a general idea of what the end of it is, okay? They don't know about the other, let's say 990 base pairs that they're trying to figure out. They know about 10, let's say. They have some prior knowledge, and they use a primer to sort of attach onto those 10 that they know. So let's imagine very quickly that they know that DNA sequencing, this single-stranded template, has the following at the 3' end. So we'll say that this is the 3' end of the DNA molecule. It has A, T, C, and G. So they're going to put a primer that they know for a fact will be T, A, G, and C with a 5' end. So this is going to be the primer, okay? They know that this 3' end, this is our known information because look, short stretch of nucleotides of base pairs known at the 3' end. So we'll say at 3' end of template. Okay, so this is our known sequence. We know that this is going to be our primer then. This is a good way to get things started. We know DNA polymerase is there because it adds nucleotides. So we'll say adds nucleotides. So NUCS will be for nucleotides based off of our primer giving it a little bit of help. What about these 4-deoxynucleotides? These are actually just a regular part of DNA. So they're just a regular part of DNA. Meaning that these are just the A's, the T's, the C's, and G's that are simple exactly how they are found in DNA. So we'll say this is actually a regular part, REG for regular part of DNA. This is where it gets a little tricky. The 4-dideoxynucleotides are actually going to serve as a fluorescent label of sorts. Okay, we're going to get to that in just a second. This is going to be critical in determining our gene sequence. So this is our first step, to denature DNA. And once we've done that, we mix it up in this concoction of things, and then we're going to actually do step 2, which is put all of this into a fancy machine of sorts. So we'll say machine. Step 2 would be to go to the machine, the sequencing machine, and start the following process, in which the machine exactly is going to synthesize new strand, okay, the machine will do this for us, strand at the 3' end, because this is something we know. This is where the primer is. The primer that we mixed in is going to go to the single strand and attach on, and the machine will synthesize a new strand starting at the 3' end, because that's where the primer is. DNA polymerase will come in and will also help the machine with this synthesis of the new strand. Now, this is where it gets a little tricky. This process is actually going to continue, so we're going to say continues, but it stops at a certain point until a dideoxy. Remember how there's 4 dideoxynucleotides? So basically think of them as modified versions of A, T, C, and G, but they're a little bit modified, so we're going to put stars next to them. They're not normal A, T, C, and G. They're actually dideoxy. It's going to continue until a dideoxy is inserted onto this growing strand. Now, what causes this stopping? Why does it stop actually at this point? This actually, we'll state it right over here, prevents, we'll say, further elongation of our growing strand. But this looks counterintuitive. This seems like, why would we do that? We want to grow and determine a gene sequence. Well, the reason why it prevents the further elongation is associated right over here. We're going to write this down, there's no free 3' OH. Who is very perturbed and bothered by the fact that there's no free 3' OH and you've just shut me down? DNA polymerase. DNA polymerase needs this 3' OH, so we'll say needs right over here, 3' OH. If you're missing it as a dideoxy and you get attached onto this new strand, DNA polymerase comes in and says, whoa, what's going on here? There's no 3' OH for me to work off of. I'm just going to stop. And this is going to prevent further elongation. We're going to see the purpose of this as we move forward. Now, at the sort of end of this process, we're going to have what are known as a set of labeled strands. Labeled strands. Now, where does this labeling come from? Remember, dideoxys are fluorescent. They shine, okay? They're very easily seen. These labeled strands are going to be of various lengths because you're going to have many different lengths due to the terminations that happen based on termination. So what if there was, an A that was early on? That A will be stopped and terminated. Let's say that growing strand will be terminated by a complementary T dideoxy. And let's say all the way down, maybe in a different strand that's within this concoction, there's going to be a G. That's going to be stopped by a dideoxy C. Whenever it shows up, that G shows up, you're going to have a dideoxy C right next to it. This is going to cause a variation in lengths based off of the termination that we've established. Now, we're going to actually get some very important knowledge due to color, meaning that the color that is seen on these labeled strands, these fluorescent labels, will show a color and it will define the last nucleotide that was inserted. This is a critical component. It tells us which nucleotide was definitely inserted. And in the final step, what we're going to do is step 3, which would be to run on what is known as polyacrylamide gel. So we'll say run on polyacrylamide gel. So we're running a little short on time. We're going to continue this in a part 2 of this DNA sequencing video, and then we'll conclude DNA sequencing with some final thoughts.